Welcome to Cell Biology. Today, we are going to explore vesicular transport, the fascinating delivery system that keeps our cells alive and functioning. We will learn how cells package materials, why they cannot rely on simple diffusion, and how motor proteins act as tiny engines to move cargo exactly where it needs to go. To understand how this works, imagine a bustling city with factories producing goods. If workers just threw goods out the window, it would be chaos. Instead, goods are packed into trucks and driven along highways. In a cell, the goods are proteins and lipids. The highways are cytoskeleton tracts, and the delivery trucks are tiny bubble-like structures called vesicles. This entire process of packing, shipping, and delivering cellular cargo is what we call vesicular transport. It is an active, energy-requiring process that is absolutely essential for moving large molecules or bulk quantities of smaller molecules safely across the cell. Now that we know what vesicular transport is, let us look at how it actually happens. The process begins with the formation of a vesicle, which is a small, spherical sac enclosed by a lipid bilayer. This sac pinches off from a donor membrane, like the endoplasmic reticulum. By encapsulating the specific cargo molecules, the vesicle prevents them from mixing with the general cytoplasm. Once formed, the vesicle does not just float away. It is actively transported by motor proteins that walk along cytoskeletal filaments. Finally, it reaches a specific target membrane, like the Golgi apparatus, where it fuses and releases its contents. Let us visualize this entire process with a diagram. On the left side of your screen, you can see the blue donor membrane, which represents the endoplasmic reticulum. Notice how it curves to form a budding vesicle, trapping the red cargo molecules inside. Once fully formed, this vesicle attaches to a green motor protein, specifically kinesin. This motor protein acts like a tiny walking machine, moving steadily along the gray microtubule track. Following the arrows to the right, you see the final step. The vesicle reaches the target membrane, representing the Golgi apparatus, where it fuses and successfully delivers its cargo. Seeing this pathway in action brings us to why it is so crucial for our survival. Without vesicular transport, complex eukaryotic life would be absolutely impossible. For instance, our nervous system relies entirely on this process to release neurotransmitters. These are the chemical signals that allow our brain cells to communicate with our muscles. Similarly, when you eat a meal, your pancreas uses this exact same vesicular transport system to secrete massive amounts of insulin into your bloodstream, which regulates your blood sugar. If this logistical system slows down or fails, it leads to severe diseases, including neurological disorders and metabolic syndromes. While this system is essential, you might wonder why the cell uses so much energy to build vesicles instead of just letting molecules float freely to their destination. To understand this, we have to look at the physics of molecular transport. The alternative to active transport is simple diffusion, where molecules move randomly. However, the time it takes for a particle to travel a certain distance by random diffusion increases with the square of the distance. This means diffusion is incredibly fast over tiny microscopic distances, but it becomes hopelessly slow over long distances, making it useless for sending signals down a long nerve cell. To understand this limitation, we can look at the mathematics of diffusion. This formula calculates the time required for one-dimensional diffusion. The variable T sub diff represents the time required for a molecule to diffuse, measured in seconds. The variable x represents the distance traveled, measured in meters. Finally, d is the diffusion coefficient, measured in square meters per second, which depends on the molecule's size and the fluid's thickness. Notice that the distance x is squared in the numerator. This means if you double the distance a molecule needs to travel, the time required does not just double, it quadruples. As distance increases, the time required skyrockets. Before we move on, let us see this with a concrete example of a 1 meter long motor neuron in your leg. The cell body needs to send neurotransmitters to the end of the axon. We're given a distance of 1 meter and a diffusion coefficient of 1 × 10⁻12 square meters per second. We substitute these values into our diffusion equation. Squaring the 1 meter distance gives us 1. Multiplying the denominator gives us 2 × 10⁻12. Dividing 1 by this tiny number yields a massive result. The final answer is 5 × 10⁻11 seconds. This is the time it would take if the cell relied purely on random floating. Since diffusion is clearly too slow, cells use motor proteins, which brings us to the active transport formula. This is an active, directed process driven by ATP energy. The formula is a simple linear relationship, much like a car driving down a highway at a constant speed. Here, T sub active represents the time taken to reach the destination in seconds. The variable x is the exact same distance to the target in meters. The variable v represents the constant velocity of the motor protein carrying the vesicle, measured in meters per second. Unlike diffusion, this relationship is linear. If the distance doubles, the time only doubles. And as the velocity of the motor protein increases, the delivery time decreases proportionally. Now let us apply this linear formula to the exact same motor neuron. We still have a distance of 1 meter, but this time a kinesin motor protein is carrying the vesicle at a constant velocity of 1 × 10⁻⁶ meters per second. We substitute the distance of 1 meter and the velocity into our active transport equation. Dividing 1 by 1 millionth gives us exactly 1 million seconds. This is a straightforward linear calculation because the speed of the motor protein never changes. The final answer is 1 × 10⁻⁶ seconds, representing a steady walk along the microtubule track. So, we have calculated both times. This brings us to a mathematical proof of why active transport is mandatory. Let us compare the results. The active transport time is 1 million seconds, which is about 11.5 days. While this might seem slow, let us look at the diffusion time. 5 × 10⁻11 seconds is roughly 15,800 years. If your nerve cells relied on diffusion, it would take thousands of years just to wiggle your toe. This massive mathematical difference perfectly illustrates the physical limits of random molecular motion. It proves that the active, motor-driven vesicular transport system is an absolute requirement for the survival of large, complex organisms. Now that we have proven why vesicles must be actively transported, let us look at the physical capacity of the delivery truck itself. Vesicles are incredibly efficient at bulk transport. A typical synaptic vesicle forms a perfect microscopic sphere. The total internal volume of this sphere dictates exactly how much neurotransmitter cargo it can hold. Understanding this volume helps us see how efficient bulk transport truly is. Because of the geometry involved, even a tiny change in the vesicle's radius can drastically change how much cargo it can carry. This is exactly why cells carefully regulate vesicle size to ensure they are delivering the right amount of chemical signals. To find out how much cargo a vesicle can hold, we use the standard geometric formula for the volume of a sphere. In this formula, V represents the internal volume of the spherical vesicle, measured in cubic meters. The symbol pi is the standard mathematical constant, approximately 3.14159. Finally, R is the radius of the vesicle, measured in meters. The most important feature of this formula is that the radius is cubed. This cubic relationship is fundamental to cellular packaging. It means that a small increase in the size of the lipid membrane will result in a massive, exponential increase in the amount of cargo the vesicle can hold. Let us put this formula to work by calculating the volume of a typical synaptic vesicle. We are given a radius of 20 nanometers, which is 20 times 10 to the negative 9 meters. We plug this radius into our volume equation. First, cubing this radius gives us 8 times 10 to the negative 24 cubic meters, representing the three-dimensional space. Next, we multiply this tiny value by the constant 4/3 pi, which scales it to the exact shape of a sphere. The final answer is approximately 3.35 times 10 to the negative 23 cubic meters. This volume is large enough to pack in several thousand neurotransmitter molecules, creating a strong, instantaneous chemical signal upon release. We have covered a lot of ground today, so let us review the most important ideas. First, vesicular transport is the highly organized, energy-driven logistical network of the cell. Second, by packaging cargo into membrane-bound vesicles and actively driving them along cytoskeletal highways, the cell overcomes the severe distance limitations of simple diffusion. Finally, this system ensures that vital proteins, hormones, and neurotransmitters are delivered exactly where they are needed in bulk quantities. Understanding this process is the key to understanding how cells maintain order, communicate, and sustain complex life. You now have a solid foundation in cellular transport.